annotation instructional video. So the first module is the basic information module. So after logging into GENI-ACT in your account, you should see one gene. So click on that gene and that will take you to the lab notebook. So at the top of the lab notebook, what you'll see is the organism. This is the organism of which contains the gene that you are annotating. And below you will see the locus, or basically the name of your gene, which is MTH928. It's actually not a name, it's more of a designation. And then below you'll see all the modules, and if you click here it will take you to instructions for all the modules. Now if you scroll down, you'll see the first module, which is the basic information module, and this is what we will be talking about in this first screencast. So the first thing they ask for is DNA coordinates, or basically where is the gene located within the genome. And to get that information you need to go to the gene page. So click on the gene page, or control click if you're on a Mac, and that should take you to the gene page. It will actually open up a new tab and take you to the gene page. Okay, so this is what the gene page looks like. So here is your gene in yellow. Whatever gene you get, it will be marked in yellow. So look at the gene and look at which direction it is going in. So look at the head of the arrow. So my gene is going backwards. That means it's on the complementary strand of the DNA. Now the first thing they ask for is the coordinates of the gene. So these are the coordinates of the gene. It starts here and ends here. So all you do is copy and paste this back into your lab notebook. So here where it says DNA coordinates, you copy and paste it into your lab notebook. Then they'll ask you for the DNA sequence and the protein sequence here. DNA sequence and protein sequence. So you go back to the gene page, you should still have your tab open, and you scroll down. So once you've scrolled down, here is your DNA sequence. It will say nucleotide sequence because DNA, as we know, is made up of nucleotides. All you need to do is copy paste everything in black, the whole sequence in black. Don't copy the grayed out regions. Copy and paste that back into your notebook. Then come back to the gene page and copy and paste the amino acid sequence. And this is your protein sequence because proteins are made up of amino acids. So you basically copy the entire thing and paste it back into your lab notebook. And during genome annotation, we will mostly be working with the amino acid sequence. Okay, let's go back to our gene page and see what else they ask us for. So we've put in our DNA coordinates, so now we know where the gene is located within the genome. We've also copied and pasted our DNA sequence into our electronic lab notebook. Now it asks for the DNA sequence length and also for the protein sequence length. So you go back to the gene page, which is already open in the tab up top. And what you do is you scroll up here, and where it says length, just under coordinates, what it gives you first is the nucleotide sequence or the DNA sequence, and the second number is your protein sequence. So you copy and paste both of them separately, and you put in your protein sequence and your DNA sequence here into your laboratory notebook. And because mine is already saved, I don't need to save it, but at the bottom here what you'll see is a save button, and you should save your module. So we've just completed Module 1, which is the basic information module, and what this tells you is some basic information about the gene that you will be annotating.